Okay guys, now we're going to move on and talk about metabolism, and we're going to just talk about three main pathways across three different chapters. First we'll talk about glycolysis and some of the other pathways related to glucose. Then we'll talk about the citric acid cycle, and finally the electron transport chain. So again, all of these are examples of metabolism. So metabolism basically just means the process of making and breaking down things in cells. So if you remember way back at the beginning, we said that everything that all the molecules we've been talking about in cells are produced in those cells, either by the cells themselves, or they come in as food from other organisms. So all organisms can basically be divided into two types. We have the autotrophs and the heterotrophs. So heterotrophs can get their energy, if they're photosynthetic autotrophs like plants, they get their energy from the sun. But then in addition to energy, all organisms need a source of carbon, oxygen, and nitrogen. So for autotrophs, they get their carbon from carbon dioxide, mostly from the air, as well as water. And that's also their source of oxygen. And then they use light from the sun to convert that carbon, oxygen, hydrogen into molecules like glucose, for the most part. And then they break down those products to get that energy back out in the source of their biochemical reactions that they have to carry out. And then if you're a heterotroph, that gets its energy and matter from other organisms, then you eat plants or eat other animals, and that's how you get your carbon and other elements through your food. And then oxygen you get from the atmosphere, and that oxygen comes from those photosynthetic autotrophs, because those organisms, as they produce their carbohydrates from carbon dioxide and water, they produce oxygen as a byproduct. And then those organisms also use oxygen. So to make this little picture more complete, we would say that the organic products and oxygen that these organisms use, they also use themselves. So they are themselves part of that cycle. But for heterotrophs, again, our only source of energy and carbon comes from other organisms. But ultimately, all that matter, and there's a separate cycle for nitrogen that is a little more complicated, but we don't need to get into it. Again, the same for us, for those of us like animals that need other organisms, we get our nitrogen from our food for the most part. Again, so it means that for matter, for things like, again, carbon, oxygen, hydrogen, nitrogen, those are the four main elements. They are part of this sort of never-ending cycles. The organisms get them either from their food or from the environment to build new molecules, and then when they themselves get eaten, or when they decay, those elements move on to support other organisms. Energy, meanwhile, is not recycled that way. So energy can, is conserved, so it can either be created or destroyed. And once it's used by an organism, it is usually released as either heat or entropy, and it can't be used anymore by another organism. So energy primarily for life on Earth comes from the sun. And so again, metabolism, the reactions of metabolism in a cell, are all those reactions necessary for either storing energy and building new molecules that the cell needs, or breaking them down and releasing that energy. So the process of building up molecules and storing energy we call anabolism, and then the process of breaking down energy containing nutrients and producing byproducts is called catabolism. So metabolism is a combination of those two types of reactions. So again, for building up molecules, anabolism, You start with, most cells, at least animal cells, start with precursor molecules like individual amino acids, small sugars, mono and disaccharides, simple fatty acids, and then the nitrogenous bases, and then using chemical energy mostly from ATP, we make larger macromolecules like proteins, polysaccharides, lipids, and nucleic acids, and then also all the other things that cells need energy for, like moving and transporting things across the membrane and so on. And then catabolism consists of taking energy containing nutrients like carbohydrates, fats, and proteins. Again, if you're a heterotroph, you get those from your food, and then breaking those down into their constituent, their lowest energy end products like carbon dioxide, water, and ammonia, when you're, especially for the proteins, and extracting that energy and using it to make ATP from ADP and inorganic phosphate. And then these other molecules like NAD+, NADP+, and FAD also store energy as what we call electron carriers, and they're part of that process as well. So that's kind of what we're going to be talking about. So this first chapter deals with glycolysis. So glycolysis is the process of breaking down glucose, which is, of course, one of these energy-containing nutrients. And so what's going to happen is we're going to talk about how it gets broken down and how its energy gets used to make ATP through several types of reactions. So glycolysis, again, the word glycolysis just means glyco means sugar, lysis means to break. So it's about breaking down sugars. And most of the time when we're talking about glycolysis, we're talking about breaking down glucose, because glucose is one of the most common monosaccharides that organisms produce, and it's produced by most plants, which is where most of it comes from in your diet. So if you have glucose in your body, a lot of it came from plants you ate, although you do have the ability to create glucose, and we'll talk about that a little bit too, how you can make glucose from its byproducts. But the reason that glucose and other monosaccharides are a good energy source is that if you completely break it down, completely oxidize it to carbon dioxide, it yields almost 3,000 kilojoules per mole of energy, and it can efficiently be stored as a polymer. So we talked about polysaccharides, so you can take glucose. Glucose can be made into both glycogen in animal cells and starch in plant cells, and those molecules are densely packed with energy and have low osmolarity, and so they are nice ways to store energy in the cell. And it can be converted either directly or indirectly to precursors that can be used for other things. So again, glucose can be stored as energy. It can be oxidized and through glycolysis, as we'll talk about in a second, for energy. And then it can be converted into structural polymers too. So glycogen and starch, remember, those are the storage polymers. But then things like cellulose, for example, in plants, are the structural polymers of glucose. And so they also serve that function. And then glucose can be converted through a separate pathway that we'll talk a little bit about, called the pentose phosphate pathway, into ribose 5-phosphate, which is then the precursor for things like nucleic acids and so on. So that's the reason why glucose is probably so commonly used by so many different organisms as an energy source. So these reactions of glycolysis are found or occur in pretty much every cellular organism in some form or another. The individual enzymes might behave a little bit differently. Some of the steps there might be extra steps or fewer steps in some cases, but it's a pretty common pathway across most living things. So the goal of glycolysis is to break glucose, which is a 6-carbon sugar, so it's a hexose, into 2 3-carbon molecules of pyruvate, and some energy is captured in this process by making ATP from ADP, and by making molecules of NADH, which we'll talk about what that is later. But that's an example of an electron carrier. And then over here, this is just a summary of the steps, which we'll go through briefly, just to give you an overview. It looks complicated, but there's only really there's 10 reactions, but they're pretty straightforward. It's really just a process of converting the glucose, phosphorylating it, and then breaking it into two parts, and then converting those eventually into pyruvate. So there's two phases, as it's kind of broken down here. The first 5 steps we call the preparatory phase, which is basically about phosphorylating the glucose and breaking it up into 2 molecules of glyceraldehyde 3-phosphate. And then the second half is called the payoff phase, where the glyceraldehyde 3-phosphate is oxidized eventually to pyruvate, and that produces the ATP that this reaction nets out. So we'll talk about the preparatory phase first. Again, the preparatory phase is basically about sort of getting the glucose ready to be oxidized and to extract ATP from. So it mostly involves phosphorylating the glucose. There's 2 phosphorylation reactions, and then breaking it into 2 molecules of glyceraldehyde 3-phosphate. And then again, this is just summarizing the payoff phase. So we're taking that glyceraldehyde 3-phosphate, oxidizing it eventually into pyruvate, and then getting ATP from it. And also NADH is produced as, again, an electron carrier. So the first step, again, is converting, or the priming, during the priming phase, is phosphorylating glucose. So glucose gets phosphorylated in the first reaction on number 6 carbon by an enzyme called hexokinase, and so this requires a molecule of ATP. And so this is what the reaction looks like up here. So you just hexokinase basically adds a phosphate to the number 6 carbon up here. The purpose of phosphorylation probably it for one thing makes it harder for glucose to actually leave the cell. So phosphorylated glucose does not go through the glucose transporters that normally move glucose in and out of the cell. So that's probably why phosphorylation, or one of the reasons why phosphorylation happens as one of the first steps of glycolysis. And then it also kind of gets it ready for the subsequent steps. So a lot of the enzymes that are downstream from this reaction have binding sites that require phosphorylated glucose or phosphate groups to bind. So now down here, this actually, again, this is all from your book, but this is just a little picture where they've sort of simplified the structure of the molecule into sort of linear form. So again, this is what glucose looks like in its circularized, cyclized form, and here they've sort of stretched it out into a straight line with each of the carbons just represented by a little branch point along this line. And you can see the OH groups. Of course, glucose is an aldehyde sugar. So when it is linearized, that number 1 carbon is in its aldehyde form. So again, all that happens in the first step is that a phosphate group gets added to that number 6 carbon. So this is now glucose 6-phosphate that we've produced here. And then the second step just involves an enzyme called phosphohexose isomerase, because all you're doing is isomerizing glucose into fructose. So fructose is a ketone sugar. So all we're really doing is moving the carbonyl group from the number 1 carbon to the number 2 carbon. And again, that's probably important because it allows the next step to happen. It also means, of course, that again, if you had fructose. There's also enzymes that phosphorylate fructose, so you could have fructose itself as an energy source instead of glucose. So that's another thing to point out, that even though we call this glycolysis, we start with glucose as our precursor, there are other molecules that could be the starting point for this reaction. So now we've got fructose 6-phosphate, which again is just this is what it looks like linearized. So we've just moved the carbonyl oxygen from the number 1 position to the number 2 position. And then we have another phosphorylation step. So another kinase. This one's called Phosphofructokinase because it takes fructose 6-phosphate and adds another phosphate to the number 1 carbon. Now we have fructose 1,6-bisphosphate. Again, this is why isomerizing to fructose was necessary, because now we've got 2 phosphates, whereas these internal carbons are harder to phosphorylate than the ones on the end. And so by converting the number 1 carbon to have a hydroxyl group instead of a carbonyl, now there's another spot that can be phosphorylated. And that also means that now when in the next step, when this gets broken down into 2 3-carbon molecules, both of them will be phosphorylated. And that's what happens next. So now we have an enzyme called aldolase, and aldolase basically just cuts this molecule, fructose 1,6-bisphosphate, into 2 3-carbon molecules. So dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And so that's what's going on down here too. The enzyme breaks up. The reason it's called aldolase is because dihydroxyacetone is a ketone, and then glyceraldehyde 3-phosphate is an aldehyde. So that's how you ended up with that name. But the idea is now we have 2 3-carbon molecules, and they can be converted into each other, and that's what happens next. So then an enzyme called Triose phosphate isomerase takes dihydroxyacetone phosphate and converts it into glyceraldehyde 3-phosphate. So that just involves sort of converting the OH group here on the end of dihydroxyacetone phosphate into a aldehyde group, and then the internal carbon has an OH group on it. So now we have just 2 molecules of glyceraldehyde 3-phosphate, and so now they can both go through the rest of the reactions through the same pathway. So again, those are the priming steps. And so that just gets everything ready now for the last 5 steps, which is where we start to oxidize these molecules and start to actually get some energy out of them. So the first oxidation step involves taking glyceraldehyde 3-phosphate and adding another phosphate to it to make it 1,3-bisphosphoglycerate. But this is not done with ATP. Instead, we use inorganic phosphate, and then a molecule of NAD+. NAD+ is, so NAD stands for nicotinamide adenine dinucleotide, and we'll talk about it more later because it'll be really important later when we get to talking about the electron transport chain. But this enzyme, glyceraldehyde 3-phosphate dehydrogenase, adds that phosphate group, but in the process reduces NAD+ to NADH. And so that basically actually takes some of the energy stored in this and uses it to... So the next step then is the production of our first ATP molecule, which the second phosphorylation or oxidation of glyceraldehyde 3-phosphate was necessary for. So now we have an enzyme called Phosphoglycerate Kinase, which will take this bisphosphoglycerate that we made last time and a molecule of ADP, so this is ADP here, and it takes that phosphate from from here and moves it over to ADP to make ATP. And so that energy then that was used to add the phosphate in the first place is then used to make now ATP. So this is our first ATP that we've made that actually can be used now by other enzymes that require energy. And now we're left with 3-phosphoglycerate as the next intermediate. Now you may wonder, this enzyme is carried out by a kinase, but in this case the enzyme is actually removing a phosphate from something, whereas kinases we normally think of as enzymes that add phosphates, but this enzyme does add phosphates to phosphoglycerate in the reverse reaction. So it's just when the concentration of this phosphoglycerate is high and the concentration of ADP is high and ATP is low, that it moves in this direction. So that's a property of a lot of enzymes, is they're capable of driving their reaction in the opposite direction, just depending on the relative concentrations of their products and substrates. So now we have 3-phosphoglycerate, and the next step, there's still one more ATP we can make, but before we do that, there's an enzyme called phosphoglycerate mutase that will basically just move that phosphate from the end carbon here to the internal carbon, and that gets the reaction, or gets the now we have 2-phosphoglycerate, which is now ready for the last couple steps, which first we through a dehydration reaction, meaning removing a water molecule from 2-phosphoglycerate, introducing a double bond. Now we have phosphoenolpyruvate, and that dehydration then gets us ready for to allow the phosphate, that last phosphate, to be transferred to another molecule of ATP. So that's what's that's basically the last step, is pyruvate kinase takes this phosphoenolpyruvate, breaks off that phosphate, again transfers it to ATP, and that energy then gets transferred. So now we have another molecule of ATP, and that is then able to be used by other processes, and then a molecule of pyruvate, which we'll talk about more later. It'll go on into other reactions. This just to show kind of the balance sheet of what we started with and what we ended up with. So again, for each glycolysis reaction, you start with one molecule of glucose. You use 2 ATP molecules. So that might seem counterintuitive. If the point is to make ATP, why are we using ATP? But again, those two phosphorylation steps at the beginning, priming the molecules, involves phosphorylating them, and so that's where the ATP shows up. 2 molecules of NAD+, which are again going to be electron carriers, 4 molecules of ADP, and 2 inorganic phosphate groups. And then what you get out of the reaction at the end is 2 pyruvates, 2 molecules of ADP, which were broken down from ATP, 2 molecules of NADH, which are the reduced forms of NAD+. Those are going to be used later in the electron transport chain. 2 protons, 4 ATPs. So if we back up, remember that we have 2 ATP producing steps, one here by pyruvate kinase, and one here carried out by phosphoglycerate kinase. So both of those reactions produce ATP, but remember that from one glucose we have 2 of these guys, 2 bisphosphoglycerates, and 2 phosphoenolpyruvates. So you end up with a total of 4 ATPs getting produced. So the net number of ATP molecules made is 2. So we used 2 to carry out the priming steps, but we produced 4 in the payoff phase. So you net out 2 molecules of ATP for every 1 molecule of glucose, plus 2 NADHs, which again will be used further down the line. So we did net out more energy than we put in. And again, pyruvate is not the end of the story. So pyruvate is the end product of glycolysis, but then pyruvate can be broken down further into other products, and more energy can be broken down, basically depending on whether oxygen is present or not. So in yeast and in other single-celled organisms, pyruvate can be broken down into, or oxidized further, or not oxidized, but broken down into ethanol or carbon dioxide. This process is called fermentation. In fact, this is where this is how things like bread and beer are made. So if you make bread, if you put yeast in with a sugar source like glucose or flour, it will convert it into ethanol and carbon dioxide. The carbon dioxide forms little bubbles, and that's what makes the dough that leads to your bread rise up and be bubbly. And then if you do that in liquid form, it will form beer or wine or whatever beverage you're trying to make. Now, in animals, in hypoxic, meaning low oxygen conditions, or anaerobic, which means completely in the absence of oxygen, pyruvate can be broken down into lactate or lactic acid. And this is actually the case in your body. So whenever you have muscles that are vigorously contracting, meaning that you're working your muscles, they're using energy faster than they can be resupplied with oxygen, your muscles will start breaking down pyruvate into lactic acid, which is why if you work out real hard, your muscles eventually start to burn and feel sore. That's because of the buildup of lactic acid in those cells. In aerobic conditions, meaning in the presence of oxygen, pyruvate gets broken down further into acetyl-CoA, and then it goes through a whole another set of reactions called the citric acid cycle that eventually breaks it all the way down into carbon dioxide, and then that energy is used to produce more and more ATP. So we'll talk about those reactions more later. So now, before we move on from glucose, because now we've talked about how glucose is broken down, but it's worth pointing out how glucose is made. So this is called gluconeogenesis, which just means formation of new glucose. And so, of course, in plants, glucose is made mostly through the process of photosynthesis. So plants take carbon dioxide from the air and using energy from the sun, using 3-phosphoglycerate as a starting point, or they convert CO2 into 3-phosphoglycerate, and then that gets turned into glucose 6-phosphate, which then can be converted into starch or sucrose or other sugar molecules like other disaccharides. It can be used to make glycoproteins and so on. And then again, that's how the plants store energy, or they use those molecules as for whatever purposes they need. And then both plants and animals can take molecules like triacylglycerols. Remember, those are basically fat molecules, and the glycerol part can be converted into glucose 6-phosphate by using energy. And so then that's one of the ways in which glucose can be made by animal cells. But then there are some glucogenic amino acids. So those are just amino acids that can be converted into glucose. So they enter through the citric acid cycle, and then they can be converted into phospholynyl-pyruvate. And then that can be converted basically in a reaction that looks a lot like backwards glycolysis back into glucose 6-phosphate. And then again, in animal cells, they can be converted into other monosaccharides or disaccharides. They can be converted into or used to make glycoproteins, or for storage, they can be converted into the storage form of glucose in animal cells, which is glycogen. Or they can just be present in the blood as blood glucose. And then the other way that animal cells can make glucose is by taking lactate, converting it back into pyruvate, and then again it sort of through a reverse of the glycolysis process, convert it back into glucose 6-phosphate. We don't need to worry about all the individual steps of those. I just wanted you to see kind of an overview of how this happens. And then the last thing to say about glucose is about the pentose phosphate pathway. So apart from glycolysis, this is probably the second most important reaction cycle that glucose goes through. So phosphorylated glucose, glucose 6-phosphate, which again, that's the first thing that gets produced in glycolysis. And if it doesn't go through glycolysis, it can go through this other set of reactions that eventually leads to ribose 5-phosphate. So glucose 6-phosphate gets oxidized to 6-phosphogluconate, and then get oxidized again to ribulose phosphate, and then that gets converted into ribose 5-phosphate. Of course, ribose 5-phosphate should sound familiar because it is the sugar part of nucleotides, and so all the nucleotides have a ribose group on them. With a 5' phosphate attached, and so all the nucleotides, all the DNA and RNA molecules need ribose 5-phosphate. There are a lot of other molecules, a lot of coenzymes that need ribose 5-phosphate too, and that's where that comes from. This reaction does produce some energy, so these two oxidation reactions use NADP plus as an electron carrier. And then NADPH, which is the reduced form, is an important part of other reactions. So, for example, this enzyme called Glutathione Reductase is an enzyme that breaks down this molecule here called glutathione. It's GSSG. The S is for sulfur, so it basically has this disulfide bond inside of it. And so it gets reduced by this enzyme called Glutathione Reductase. Using an ADPH, this enzyme is really important for removing oxygen radicals from the cell. So these glutathiones, in any case, are an important sort of antioxidant in the cell. So that's a reason why you want NADPH present. And then NADPH is also used by a lot of enzymes that make things like fatty acids. So the enzymes that go through the reductive biosynthesis of those molecules need NADPH also, so that's where that one comes in. All right, and so that is all we're going to talk about for glycolysis. Next, we will talk about the citric acid cycle.